So I'm very excited to come here today and give all of you an update. But I'm particularly happy to see all of my old P41 colleagues here who had to listen to me for so many years. I'll give a very brief background for all the younger people here, but then jump right into kind of all the new things that have been happening since I went to Boston University. Listen here. Just to start us off, fMRI, of course, has taught us a huge amount how the brain works. It has helped us measure brain responses to see how we respond to motor sensory stimuli, how we respond to language, how we compute, and in a very limited way interact with others. Should I mute the other people? You. Okay. But it has many limitations. A person has to be lying down in a magnet, and that really kind of limits the natural behaviors that we can measure a subject engaged in. And it has some limitation on the populations that you can study. So that's where FNIRS and EEG can help, because they can allow a subject actually to be walking around the everyday world. We can be measuring natural behaviors. FNIRS is measuring the hemodynamic responses to neural activity, just like fMRI, and of course EEG is measuring more directly the electrical neural activity. They are being integrated together as wearable systems. It's really still early stages of these wearable systems, and there's more optimization happening. And of course, as we go into the everyday world and want to measure brain activity in the everyday world, we need sensors telling us what the subject's actually doing in the everyday world. And so eye tracking, IMUs, those are going to be important, measures of behavior that we can include and integrate with EEG and FNIRS. And so all this technology exists, and what cognitive scientists keep telling me is, how are you ever going to analyze that data you collect in the everyday world, right? And so that's really kind of going to be the biggest question, is how are we going to analyze this data? The technology exists, and it's getting better and better all the time. We can collect the data of human brain function as people are engaged in everyday activities, but how are we going to handle it? I'm not going to answer that question today, but I am going to give you kind of an update of the types of data that we can get and our beginnings at how to analyze those data. Okay, this is a slide that the P41 crowd listened to me for 15 years about neural activity, neurovascular coupling, right? EEG is measuring that neural activity directly. It releases vasoactive agents, neurotransmitters, that diffuse to the arterial wall, relaxes them, blood flow increases, blood volume increases, it delivers more oxygen to the tissue. At the same time, the membranes of the neurons have to repolarize, it needs more ATP, consumes more oxygen. So we have this competition in the oxygenation of the hemoglobin, but for sure blood flow is increasing, total hemoglobin is increasing, and generally oxyhemoglobin is increasing as well. And we wash deoxyhemoglobin out of the venous side. And this is what you measure with bold fMRI. Okay, so that's the first 18 years that worked on at MGA. Now let's go to the future, right? So we want to go from the lab to the everyday world. So we want to be measuring brain activity, not just in a subject sitting in the lab or sitting in the Bohr magnet. We want to be measuring brain activity as they're walking down the street, interacting with other people, and perceiving the world around. And so at BU, I have many colleagues who are interested in this for many translational applications, including Parkinson's disease and how that affects gait. You want to measure that as a person is kind of walking around the world and see what is going on in the neurodegeneration that's affecting the gait and the quality of life. Stroke also affects people's gait or language. So we're looking at aphasia as well. And also, I'm very interested in perception and attention and working memory. Some colleagues on that. So back to the first 18 years, this was our first system. This actually helped me get the job here. I remember coming to my interview, bringing this system, and with Greg Sorensen, and we were trying to measure finger tapping activity, I think, in Greg's office. Then we got here to MGH, we started evolving the system, started working with a company, built this nice system, imaging system. I think we published 50 papers of brain activation with that system, and then we built that last system and probably published 100 papers with that system, and actually sold about 100 of those around the world. to other people as they were measuring human brain function with these systems. So already by 2004, kind of the hardware was way ahead of the experiments, and so we stopped developing hardware, and we just focused on the methodology for analyzing the data and doing experiments. Others, however, continued to work on hardware. And it's starting around 2012, we started to see more and more wearable systems. And basically that's because electronics were getting smaller and smaller, more powerful. And so people were making more and more wearable systems. I was still focused on data analysis. I was trying to, there was much more data analysis to do. But by 2014, 2016, I kept getting hammered by reviewers that would not fund my grants trying to advance data analysis methods. And that was making me very sad. So I decided, oh, let's just throw in a name where we're going to make a wearable system. And so I resubmitted a grant where all I did was add a name saying we'd make a wearable system, and I got a 10, which is a perfect score. So I was very happy about that. And that started a whole new life for me in FNIRS, where we moved back to hardware development. So this was our initial idea. We built an optode. This was like maybe 20 millimeters, 24 millimeters in diameter, had a source and detector. We kind of built that. But in the process, we really made them even smaller. So we could make the sources and detectors smaller than a penny. And so we wrote a nice big Brain Initiative UO1, where we were going to make a whole head system with individual sources and individual detectors. We were going to make this high density. We wrote that in 2019, and in 2020, it actually got funded for submission, so I was very happy about that. And the vision was we were going to build this wearable, high-density whole head system with, I think we were saying 144 optodes on the head, and we were going to have eye tracking, and we were going to try to decode people's brain activity as they were interacting with the world. That was the vision. Okay. Then, of course, COVID happened. And Alexander von Lumen, who was my postdoc at the time, he helped me write this grant. He went back to Germany. He actually went to go work with NIRX. He became their chief technology officer. NIRX was the biggest FNIRS company presently. He sent me one of their systems, one of their wearable systems. It looks like this. It has 16 sources, 16 detectors. So way less than what I want to have. You could actually have 4 of these and it'd be 64 by 64, which is still less than what I wanted to build. But it would be 4 of those and it'd be pretty heavy. So we basically wanted to drive, right now we're driving 200 optodes with a box that's that size. Right? But anyways, we were in the early stages of let's build this, and he sent me their system. It was COVID, it was like December, and I had this system at home. So it was a really nice Christmas present. I said, well, I have been trying for, almost 15 years to do a high density measurement. Like all FNIRS, today, the majority of FNIRS, you have sources detectors 3 centimeters apart. But if you can actually get a higher density array of measurements, you'll get better spatial resolution. You can improve your spatial resolution by a factor of 2. And so I said, let me try to do that with the system. And so you can see these are the grommets that are going to hold the optodes. So I put the optodes in those grommets. I put them over my motor cortex, because the first thing we always do is a finger tapping task. And I got a high density, high resolution image of finger tapping in my brain, at Christmas time. So I really enjoyed that. We've been trying with our own system for over 10 years to do this, and just the hardware was too annoying, and we were not able to do it. So I was very excited that I got this system on loan from NIRX, and they had this system actually for like 4 years, and nobody had ever done a high density measurement. So they didn't even know they could do it. It was kind of interesting. And so we were able to show that. So we said, this is great. This is exactly what we want to do, but we want to cover the whole head. And so we set out doing that. Let me just do a little plug. Maybe you saw that 3D printed cap. We were, for years, we had software, we call this Atlas Viewer. We would design where the optodes go on the head. We would then take this fabric easy cap and we'd cut holes in that fabric easy cap exactly where those optodes are, the sources and detectors. And then we'd manually put everything in. We kept thinking, this is silly. We have a computer representation of where we want the opticals. Why do we have to manually cut the holes in this cap? And so we spent a few years, starting with a senior project and then a master's project, and we built the software that actually 3D prints these caps, just using NinjaFlex, you know, and then we ultrasonically weld these pieces together. Using NinjaFlex as material, TPU, and it works. We were wondering early on, 2021, we're still testing all the hardware ideas that we have, but Bernard and I couldn't figure out for our lives, how could we actually fit all the wires on the head? You know, 128 detectors, 32 sources, that's a lot of wires. So we built this non-functional prototype, and this was, you know, me at home during COVID, 3D printer. I kind of 3D printed all of these dummy optodes and attached these wires, and lo and behold, you know, it works. Like those, we actually couldn't visualize where those wires are going to go. So we had to build this dummy prototype, and it was obvious this is going to not be an issue. At the same time, we realized we can put even more on the head. So we said, all right, based on this, let's not do 128 and 32. Let's actually expand it to 144 and 56, really cover the head. This taught me a lot, right? That making non-functional prototypes teaches you a lot. And so that's something we're doing more and more. And it really refines then what we actually do. This Ninja Cap, though.
David, could you give just a quick background on the limits to the spatial resolution that's determined by the detector, and you know, sensor size, you know, in terms of what's the optimal you get, you know?
So, you know, for the first 20, 30 years, and still today, people are using 3 centimeter separations on adult humans because the separation has to be large enough to get through the scalp and skull. If the separation is too small, the light that diffuses back is only seeing the scalp. So you need 3 centimeters to get into the brain. And then they sparsely cover as big of a region as they can, but the resolution is really going to be the 3 centimeter separation between the source and detector. By creating this hexagonal pattern where I have like overlapping measurements, just first neighbor and second neighbor measurements, we can get to about 15 millimeters. And if we go to ultra high density, you know, making them even smaller, higher density, you can get to about 10 millimeters spatial resolution just on the cortical surface near the scalp, near the skull. So So we are actually, if my UG3 now converts to a UH3 in August, even though we met all milestones, we're going to make a cap that has 400 optos. And that would push us probably from 15 millimeter resolution to 10 to 12.
You have the same source detectors, 3 centimeter difference, just kind of overlapping.
In this case, these are 18 millimeters apart. Like the nearest neighbor is 18 millimeters apart, and this is the second nearest neighbor, and that's 30 millimeters apart. So 18, 30. When we go to ultra high density, this is going to be 12 millimeters.
But then do you see any brain signal when you look at that?
At 12, we don't, right? But then I don't remember the numbers. It's going to be like 12, 20, and then 25 and 30.
So you're looking, you're still looking at the pairs distant by roughly 3 centimeters. You just have more pairs, so you can hone in on where the centroid of activity is.
Exactly. And it also, importantly, for FNIRS, a lot of the physiological contamination is coming from the scalp. And actually, by getting that dense array of measurements on the scalp, it removes it all from the brain, and we get much higher quality images of the brain. Thanks.
Well, it helps because you know you just don't know how far to go, right?
Right. We get, you know, of course, it's diminishing returns. So this first step with a high density is a huge gain. Going to ultra high density, you know, a lot more technology for not as much gain. Okay. So these Ninja Caps have another great benefit. You know, FNIRS is challenged by dense hair, dark hair, not so much by skin tone, but it's hair is really the biggest issue. And because the ninja cap is open, we can actually pull the hair through more easily, and we're able to get good quality signal through a diverse array of hair types. And we actually just published this study, which looked at 120 people with different skin tones and different hair characteristics. And basically, it quantified for the first time, which everybody anecdotally knew, which is that as skin gets darker, but that's a minor effect, it's more the hair coarseness, hair color, but more so the hair thickness and coarseness that degrades signal quality. This we did with a sparse array, and we were happy to see that actually, you know, it's a great minority of channels that were not able to get good signal to noise ratio. But you know, so we have a new grant now where we're actually improving the sensitivity. It's going to move this window down to here, so we'll be able to get good signal on all subjects in the near future. I need to point out though, this was done with a sparse probe, and now that we're using a high density probe, we're kind of back to where we were 25 years ago, and we're learning how to optimize how to deal with hair, not with a sparse probe because we already know how to do that, but to deal with hair with a high density probe. And Ninja Cap and things like that will help us, and more sensitive systems will help us. Okay, so why 56 sources, 144 detectors? Part of it was the spatial resolution, but like the other thing out there, Joe Culver's group, they're the people really leading the way with high density and ultra high density measurements. They use a grid array, and a grid array has an even number of sources and an even number of detectors. I've always used a hexagonal array, and I always use the hexagonal array is because we have to multiplex the sources. You know, we need a detector to resolve the signal from all the individual sources. So we have to, the simplest multiplexing scheme is you turn one source on at a time. So if I have fewer sources, I have less multiplexing. So I kind of like this strategy because there's less multiplexing. So that was my rationale for using a hexagonal array. This is what Joe Culver does. He has more equal number of sources and detectors. He has more multiplexing. And so as a result, his duty cycle is slower. So I view that as a disadvantage. In theory, they can actually get a little bit better spatial resolution at the expense of a lower duty cycle. But I like the additional benefits I get with a higher duty cycle, which gives me a better signal to noise ratio. I'm also able to like cover the head where my second nearest neighbor is at 3 centimeters. I'm able to cover the head with a lower number of sources and detectors. And so just from a hardware perspective, it's just going to be less hardware, less weight, and it actually, you know, also helps on battery life as well. And because we want to build a wearable system where we're walking around the world, you know, battery life is important. Since I'm talking about batteries, I'll say actually the system now I'm about to show you, we can run 5 hours outside with with a 100 watt hour battery. Not that big. So pretty happy about that. So in 2021, Bernard did all the hardware testing to build the system, had all the designs figured out. So we placed the order July 2021 to build, you know, all of these, the control unit. The FPGAs were on back order for an undetermined amount of time. We designed everything with Intel FPGAs. So Bernard placed an order for Xilinx FPGAs. These are the two main distributors. That would require him to redesign and test everything. But we're like, we need FPGAs. This is UO1. If we don't meet a milestone, we lose our funding. So after 7 months, finally, Intel told us they couldn't deliver the FPGAs until 2024 because DoD was getting all the FPGAs, right? And so we started to panic. And then, like in May, Xilinx canceled our order for the same reason. Like, we have a milestone to meet, right? We're gonna lose our funding. And so we were trying not to panic. And Bernard looked online and he found that he was able to buy Raspberry Pi Picos, microcontrollers, not FPGAs, but microcontrollers. he bought them. They arrived a week later. So we had enough microcontrollers, but we didn't design the system to run with microcontrollers, right? So he had to redesign everything to work with microcontrollers. They're just not as strong as FPGAs. FPGAs, we were going to have frequency encoding as well as time division multiplexing. With the microcontrollers, we had to throw out our frequency encoding, but it worked. And I was really worried without the frequency encoding, it helps me distinguish our signals from room lights. I was really worried with just time division multiplexing that we wouldn't effectively discriminate our brain signals from room lights. But it worked. And that actually turned out to be a huge blessing because just time division multiplexing alone is way, way easier. Just the circuitry was way easier. So that actually really worked out in our favor. So this is what it looks like. This is the control board. These are the interface cards. Each one drives 8 detectors. So we have a lot of detectors, and then these are driving 8 sources each. This is what it looked like originally when it was all put together. That's 200 optodes there. We were going to build our own optodes, but we already had this relationship with Nerex. And building 200 optodes is not fun, and we actually have 2 of these systems, so we were going to have to build 400 optodes. So we were really happy that we were able to just buy those optodes, and there was no way we could build them better than the commercial vendors. So we were really happy to buy it from the commercial vendor, and of course, commercial vendor is really happy that, well, 1, we bought 400 optodes from them, but 2, we're showing that their optodes can be used in a high-density system like this. Okay, so that worked out well. This was the first time I put it on. 2 years after no, this is now 3 years after our grant was awarded. And so I was very happy. Actually, this is actually a baby carrier, so that we modified the whole system. So now, the last year, this is actually a picture from last year. We've kind of modified the system now. It's no longer on the front. We're wearing it on the back. There's the battery you can see, and it's completely wireless. So we actually, there's a Raspberry Pi on board here. It kind of serves up a web page. We can control the system just with our phone. It's really nice. It's really kind of cool. So we've actually, I'll show you in a little bit, just a study of people walking up and down Commonwealth Avenue and the brain signals we're getting from that. This is showing, though, the signal that we get versus distance. Joe Cover always shows these plots. So this is a log 10 scale. So signal drops orders of magnitude, from 18 millimeters, out to, we're getting signal out to 60 millimeters. This is at a low power setting. So we actually calibrate the LED powers to give us the amount of power that is optimized for each source detector location. So we can go from a low power setting to a high power setting, 100 times higher, and you just see everything rises up, right? So at high power setting, in some subjects, we can get out to 8 centimeters source detector distance. Then, we always start off with finger tapping. So this is an average of 3 subjects showing the brain activation for finger tapping. So nice spatial.
David, are there limits, safety limits on the high power, or, is it just battery life that you worry about?
This is the
high power seems better.
Yeah. High power is better. So this would be above the safety limit, but it's not on continuously. It's on less than 10% of the time, and maybe only 2% of the time. So actually, our new grant that we're working on now, the UG3, we go up to 1.4 watts, right? But because the duty cycle is one, what?
A lower duty cycle.
But because the duty cycle is 1%, it's no problem. So 1.4 watts, basically, that will go up now 20-fold. Or 20-fold would bring us to here. And so that's going to really help us with darker, denser hair, for instance. So I'm very excited about those high-powered LEDs. And I mean, it's funny that we were always focused on, we were talking about SIPMs. SIPMs are amazing, a lot of technology to get a tenfold improvement, which is great, but it's a lot of technology. A high-powered LED is the most mundane upgrade. We just have to change the circuitry to give us 20x more current. Not trivial, but it's mundane compared to a brand new SIPM highly sensitive detector. And we get more benefit from the LED than we get from the SIPM.
How does this compare to the photobiomodulation guys in terms of power density? I mean, there's people selling.
Yeah. I mean, so the photobiomodulation, you're right, the high power LEDs are getting there, but the photobiomodulation is going to be continuous.
Yeah.
And distributed over a large area, right? So I think we're still way below ever seeing any effect from photobiomodulation.
Also, in terms of safety, because people do that. If photobiomodulation is safe, then yours is definitely safe.
Oh, sure. I mean, heating is a real thing. You don't want to burn the scalp. So at 70 milliwatts, it'd be rare. I've done that experiment. At above 100 milliwatts, I can sometimes, when I have a small 200 micron fiber over a hair follicle, I'll feel it on my arm. We diverge. Let me skip over this. We've optimized image reconstruction. This was something I always wanted to do like 20 some years ago, but we never had the system to give us the data to allow us to optimize our high density image reconstructions. My student Laura Carlton has gone through all of this, and she kind of optimized everything. And actually there was a funny outcome. They're just conceptually, we reconstruct images at 2 wavelengths because we need 2 wavelengths so that we can then convert those absorption coefficient images to hemoglobin concentration images. So we got oxyhemoglobin, deoxyhemoglobin, 2 unknowns. We need the absorption coefficient at 2 wavelengths in order to estimate those 2 unknowns. We can either do what I just said, which is reconstruct each wavelength separately and then convert the concentrations, or we do it all at the same time. We do the image reconstruction and the hemoglobin concentration conversion at the same time. We always thought that was the best thing. It's called the best linear unbiased estimate, solve all the linear problems at the same time. Also, we liked using spatial bases because we know that the brain resolution is not so good. So instead of reconstructing vertices on the surface of the brain, let's just reconstruct an image of a Gaussian blob on the brain and a larger Gaussian blob on the scalp. We always thought that was the best thing to do. This analysis taught us, though, it's not great. We actually get huge crosstalk. So deoxyhemoglobin's going up. That's not cool. So we looked at no spatial bases, and with no spatial bases, we actually get a lot of crosstalk. We still get the hemoglobin crosstalk, but we get a lot of crosstalk on the scalp. So those large spatial, large Gaussian blobs in the scalp were great at pushing all that energy into the brain. So when we remove the spatial bases, the brain activity is actually coming to the scalp. Benefit, though, you see these error bars are smaller. So what's happening is the noise is being moved to the scalp. And so we actually get better noise estimates in the brain. Then we said, all right, let's just go back to the old way of doing it, reconstruct each wavelength separately. Now the oxyhemoglobin is decreasing. It looks way better. And then if we do no spatial bases, we get the best contrast, the noise resolution estimate of brain activity in the brain at the expense of crosstalk to the scalp. But if all we worry about is statistics, this is perfectly good. Okay. All right. So now let's go outside. Let's do something naturalistic. So our first experiment was, can we measure people interacting, measure brain activity of interaction while walking? And so our first experiment was kind of in a naturalist, responding to questions. So my colleague Swati Kiran, her group studies aphasia patients, and so they've done tasks with FNIRS where they are measuring the patients' brain responses as they respond to questions. And these are just questions like, how do you like to spend your Sunday afternoon? What is a favorite vacation taken. So they respond for 20 seconds. And so you typically do this in a lab setting. So we thought, let's do that on a group of healthy college kids initially, ask them questions in the lab, and then let's go outside and ask them questions while they're standing outside and while they're walking down the street. It's, so the probe covered the whole head. These are the brain regions that we were analyzing. This is all the group average data. It's 21 subjects. So sitting, standing, walking, can kind of see like a left-dominated response in language areas. I mean, we're getting responses everywhere. And this is channel space. This is before image reconstruction. So we can dwell on this, but we can also just jump to the image reconstruction. So we look at the image reconstruction. Now we got spatial images. This is the first 10 seconds responding to the question. So they listen to the question from 0 to 5 seconds, and then they respond from 5 to 25 seconds. And so we kind of see similarity across all three conditions. With the response peaking, 10 to 20 seconds during the response, and then this is oxyhemoglobin is decreasing once they stop answering the question. I was excited about these results because it just showed we can measure the same sort of typical language regions responding when a person's answering a question, whether or not they're walking outside or sitting in a lab inside. When we started this experiment, we were really worried about motion artifacts while walking, and the sunlight, for instance. These were things we didn't know if we could actually do the experiment. So we do see walking in our data, and what happens is when you walk, every heel strike, some of the optodes on the top of the head that are not situated well in the scalp, they bounce. They bounce a little. These optodes are against the scalp; they're not bouncing, but these bounce with every heel strike. But the signal change from the bounce is never really larger than about 4 times the amplitude change we get from the heartbeat. So, and your gait cycle is kind of like your heartbeat. So we're already dealing with cardiac signal. It's not that hard to deal with the gait artifact we're getting. So that was a pleasant surprise that our probe, it's actually what's happening, I believe, in part, is because we have such a high density array of optodes, they don't move, they don't bounce as much. Whereas like for the last 30 years, people are using sparse arrays, there's nothing holding it on the head as well, and they bounce much more and they get way more motion artifacts than what we get here. So it's actually with a high density probe, it seems to be easier to do these naturalistic experiments. Okay, so then we said, what about perception? So then we started another study where we had eye trackers on the subjects. And so we had 20 subjects walk up and down Commonwealth Ave. And so we'd measure them as they walked to the George Sherman Union, sit down and have funny interactions with random people. And then they stand up again and walk back to the lab. And so we said, how are we ever going to analyze these data? And so we decided to analyze it like it was a movie, right? So fMRI and now FNIRS, everyone is showing people movies and finding features in the movie and analyzing the brain and using those features as regressors to analyze the brain response to those regressors. So that works super well in fMRI, and it works super well with FNIRS. And so we said, well, we can do the same thing with people walking outside. They're wearing eyeglasses. It's got a scene camera. So we told them, walk down the street and look at people like you're watching a movie. The only difference is when you do a movie with fMRI, you have the same movie that every subject watched. And so you only have to label one movie and use it for all subjects. In our case, we had 21 different movies. And so we had to manually, with a lot of help from AI, label 21 different movies. We started off just running subjects inside because we had never done that. And we're just right now looking at like these 6 regressors: luminance, sound intensity, optical flow, faces, body, speech. So from the movies, you can get regressors for all of these. You put it into a general linear model. There's your design matrix. They analyze your data and you get these images. I don't want to dwell on this, just qualitatively, it generally agrees with prior fMRI and FNIRS work. We wanted to go outside, right? So our subjects are walking 10 minutes down Downcom Ave. They sit for 10 minutes and then they come back. And we do the exact same analysis. And we have IMUs as well, so we get the gait cycle. We even started developing all these advanced signal processing methods to use the IMUs to remove the gait cycle from our FNIR signals. At the end of the day, we didn't need to do it. It's really not a big deal. So we're very happy with that. Signal quality is good. This is actually what's really nice with time division multiplexing. We turn each LED on one at a time. We also use spatial multiplexing, but we don't need to go into that. But we have to have a dark state. So we frequently have a dark state where no LEDs are on, and we just measure ambient light. And we subtract that off from the signal so that we're subtracting ambient light, so that if there was too much sunlight, it's removed. But we can just look at the dark state, and basically you can see these periods where they walk under the shade of a tree, right? Or the sun goes behind a cloud. So we're able to get really nice measures of the environment that the subject is in. And it turned out we made this cap. It's not tinfoil. We thought of using tinfoil. We actually used the Decathlon tent. They have this patent pending tent material where you can have the tent set up at noon under full sun in the mountains, and it's pitch dark in the tent. So I bought that $100 tent, and I found a seamstress who made us 5 caps out of that tent. And it works really well. Okay.
So you know what they're looking at? Do you have another camera?
So the eye tracker, right, has the scene camera, right? So that was, remember this movie, we had this movie here. This is the subject walking down the street. So, and the AI is showing you the faces. That's how we get the face regressors. And you can even see the dot where the subject's looking. I think this subject's trying to ignore that guy. He's not looking. That was totally random. So yes, right, we got the scene camera, we got the regress. We haven't analyzed the gaze yet. That will be a future study. We actually analyze the gaze. Okay, so there's the regressors. One thing you do when you have your regressors, you make sure that they're not correlated, because if you had any correlations in your regressors, you're not going to get independent estimates of the different regressors. That's the correlation matrix of the regressors, so we're good. And just looking at 4 of the regressors, this is the outdoor movie, luminance, sound intensity, bodies, faces, and this is the indoor, completely different group of subjects, but qualitatively pretty good agreement. It's very preliminary, it's from last week, but we're working on the statistics, but I was really happy to see this last week. I totally, didn't, of course, you do an experiment like this, you want it to work, but you're so surprised when it actually does work. See that qualitative agreement. So I think this just is going to be such an amazing way to analyze naturalistic brain imaging data. Right? It's treated like a movie. So, when the first movie experiments were done by, I guess, Jack Gallant with fMRI, people thought it was cool, but where's that going to go? And now, 15 years later, it's just incredible how far it's going. I think this is kind of our first way of analyzing naturalistic brain imaging data in the everyday world, and I think we're going to go quite far. Okay. I have one more topic, and I think I have time. Before I go into the last topic, well, I'll take questions at the end unless there's anything. Last thing is I'm interested in attention and brain state. So brain signals in the everyday world will depend on brain state. And naturalistic behavior strongly modulates the attentional state of a subject, right? Attentional fluctuations drive many behavioral errors, for instance, distraction, vigilance, mind wandering. These states are regulated by neuromodulatory systems, like neuroepinephrine. I'm leading here. So Anna DeVore, many of you know, and I work with, and she was here, and now she's at BU. They just got this really nice paper in Press in Nature Neuroscience, where they show that neuroepinephrine, a neuromodulatory signal, modulates the hemodynamic response to neural activity. So this is in mice, and basically they have calcium data, fluorescence data of neural activity. They have fluorescence data of neuroepinephrine, and they basically show if we analyze, and they have total hemoglobin signals from intrinsic signal imaging. Right? So they predict the total hemoglobin signal from calcium. And then they predict the total hemoglobin signal from calcium and neuroepinephrine. And the model performance with neuroepinephrine is always better than with calcium alone, right? So they've done a lot more studies, but in summary, with that one slide, I just want to tell you that neurovascular coupling is not constant, right? It is modulated by neuroepinephrine very strongly. She also looked at acetylcholine. That has less of an effect. Neuroepinephrine seems to have a pretty strong effect, and she's starting to look at serotonin and dopamine as well. But we're like, okay, can FNIRS track attentional state? That's one question I have. But another question I have is, can we replicate these findings, right, that Anna has in mice? Can we replicate the finding in humans that a neuromodulatory state changes neurovascular function? And so I met Mike Esterman, who developed this grad CPT task, which is a beautiful task. CPT is continuous performance task. He made it gradual. I'll describe it in a minute. I'm excited about using this to look at attentional state fluctuations. And this is a great task for doing that because he gets a behavioral measure of the attentional fluctuations over a time scale of tens of seconds, right, during this continuous performance task. And so this time scale is ideal for FNIRS and fMRI. The task is basically a go, no-go task. He has these scenes of cities, different cityscapes, and occasional mountains. So every 800 milliseconds, another city appears, and it gradually fades from one city to another. And whenever a city appears, you press a button. So every 800 milliseconds, you're pressing a button. 10% of the time, a mountain appears, and you don't press a button. And so then the data you get from that, is basically we get the reaction time variability. And it's fluctuations in the reaction time that he has related to in thousands of subjects to attentional state. And so he has done lots of fMRI studies where he looks at this reaction time variability that he calls in and out of the zone. And he's regressed that with fMRI signals. And he can see different brain regions of when you're in the zone versus out of the zone. I'm also interested in looking at the evoked response to oddball events because based on Anna DeVore's work, when you're in the zone versus out of the zone, you're in a different neuromodulatory state. It could modulate the hemodynamic response to that oddball event. Right? So those are the two things we'll look at. This is what behavioral data will look like. So these are all the oddball tasks during a 6-minute trial. So these vertical lines are the oddball tasks. Here, they press the button when they should not have pressed the button. The circle is, it was a city, they should have pressed the button, but they did not press the button. Okay, this thin line is actually the reaction time. So you see a lot of variability in the reaction time. You smooth that, you get the smooth reaction time. And then Mike has defined when it's below the median, that's in the zone, red is in the zone, and blue above the median, you're out of the zone. So when you actually go out of the zone, you're kind of messing up, you're making mistakes, you have to engage all of your attentional resources to get back into the zone. So there's more mental effort in these blue regions because the subject is messing up and they're trying to get back into the zone. We also have pupil diameter. We're beginning to analyze the pupil diameter. The reason we have pupil diameter here is because Anna DeVore showed in her mice super strong linear relationship between pupil diameter and neuroepinephrine in the brain. So, we think that this might be another measure of the neuromodulatory state in the human. Of course, pupil diameter in human is much more complex than in a mouse. So these are Mike Esterman's past fMRI results. So blue is showing anti-correlation with the variable reaction time. So blue is kind of the regions that are activated when they're in the zone, and red are the regions that are activated when they're out of the zone. So you just took that regressor, right, and used it as a regressor for the bold signal, and he gets these images. So our first step was, do we get the same thing with FNIRS? We have 20 subjects. There's the group average of 20 subjects. Mike gave us 80 MRI subjects. We analyzed those, or 72 fMRI subjects. We analyzed those fMRI subjects the exact same way we analyze our FNIRS data, and just did a spatial correlation. And, 0.31, it'd be great if it was higher, but it's significant. I'll just attribute the 0.31 not being as high as I wanted it to be due to spatial resolution of FNIRS, and, just our brain unit sensitivity falls off exponentially with depth. And so it's whereas fMRI is uniformly spatially uniform sensitivity. Okay, so step one, we passed. I think, we're able to measure attentional state of this type reasonably well compared to fMRI. And then we went to evoked responses. So what I wanted to see here was whether or not in the zone versus out of the zone gave us different evoked responses. Mike Esterman had never actually analyzed his fMRI data for the oddball evoked response. So he asked his postdoc to do it, and his postdoc just gave it to us in these broad 17 networks. So these are really large areas of the brain. So Laura Carlton, my student, she did the same thing. So in these regions, we have reasonable good agreement, but these other regions, less so. This region we actually had no sensitivity to. Let's just turn off those regions and focus on some of these. So the colors, are the colors helpful for us? No. There's a new slide where the colors are more helpful. The colors are really difficult. Let's just focus on, let's see. The red, which is out of the zone, oxyhemoglobin, and the light blue. And so we can kind of see, this is in the zone, oxyhemoglobin, this is out of the zone, oxyhemoglobin. Same brain region, right? But different neuromodulatory state, we're getting different evoked responses. And we can analyze this in other brain regions as well, but like this is an attentional region. So I'm kind of focused on that a little bit. Anna DeBoer is excited that has more undershoot. There's kind of a post-stimulus vasoconstriction that she sees in our mice. So that's something we're going to be looking at. We also, now on 4 subjects, also have EG. And so far, at least in those 4 subjects, we're not seeing any change in the evoked electrical response in the zone versus out of the zone, right? So that's just lending support to these differences are not driven by the electrical activity. So neurovascular coupling in that sense is changing, right? Because the driver, which we normally think of as neural activity, is the same. Okay, so what has changed? We believe it's the neuromodulatory state, right, that has changed.
But you've shown that the behavior is different in the in and out of zone, presumably?
Well, the behavior is at reaction time.
Yeah, and so that's, by definition, it's different. So that's why it's a little surprising that there's no electrical correlate to the behavioral change.
Fair point. Fair point. So maybe it's the EEG, not a good spatial resolution measure, right? So it's just getting the oddball event.
That's just not sufficient. So
it may not be not sufficient.
So much for people that go back and talk about the electrical signal, the spiking, et cetera, as the be all and end all, and everything is that. Well, I would suggest otherwise.
Now, of course, Mike gave us 72 fMRI data sets.
Yeah.
Right. Way better spatial resolution. We could analyze that to see if there is different spatial regions that are showing different responses because of the variable reaction. Well, yes, absolutely interesting. Okay. All right. I'm out of time. Just you know, we have many other studies going on with colleagues at BU. Great range of topics, so watch those as they develop. And I'll just end there and have discussion if there's time left.